All right, everyone. I think we'll make a start. So, welcome to Combating Disease. For those of you that don't know me, my name is David, so I'm the lecturer for immunology. And my sort of research interests have been in immunology, but also virology, and looking at how Epstein-Barr virus can lead to the development of lymphoma. So what we're going to do today is we're going to have a brief introduction sort of to the module, and then we'll also have a look at the innate immune system, and then we'll develop on that over the next few weeks. So first of all, let's start off with the module introduction. So these are the learning outcomes that you can expect to know at the end of this module. So you'll be able to display a sound knowledge and understanding of the major players of both the innate and the adaptive immune response. You'll also be able to see how they coordinate and are regulated in order to defend the host. You'll also be able to discuss the mechanisms of action and also the use of a variety of drugs in the treatment of diseases with an immunological basis. And then through the practical that's associated with this course, you'll also be able to gather, analyze, and interpret data from an experimental immunology setting. So these are the topics that we're going to cover in the module. So they're broken down by the weeks. So everything in blue is sort of the immunology basis. And then you'll see that that's in the first half of the module. And then in the second part of the module, we'll sort of have a sort of pharmacology slant to some immunology-based diseases. And then you'll notice in red, we have the practical sessions. So there are actually 4 practical sessions spaced over week 5, 6, 7, and 8 that you need to attend all of them. In terms of the assessment, the actual practical session comprises 30%, whereas the end of module exam is 70%, and that'll be a computer-based exam. Then I put here some skills that you can expect to gain from the practicals, but also from the course as well. So it's important to remember that with the course, we expect you to do some self-directed study, and so independent self-study and self-directed learning. So you'll be gaining that as a skill, as well as planning and meeting deadlines, and also extracting and seeking relevant information from a whole variety of sources, including articles and PubMed as well. So I mentioned the laboratory session. So there are actually 4 parts to this. So there'll be part 1 in week 5, and then there'll be an associated post-lab the following week. And then in week 7, there'll be part 2 to the practical, and then the following week there'll be an associated post-lab. There'll be a morning session and an afternoon session, so you'll be allocated to one of those laboratory groups. And within that, you'll be assigned to a randomized group of 3 students. And it's important that you stay within that group all the way through, okay? Because you'll be working with the same samples as you move along. So it's really important to stay within your group and within your associated session. And that information will be made available on Canvas relatively soon. Again, I'm sure you're probably aware of this, but the module is supported by an associated Canvas page, so you'll be able to find all the information on there. It's also broken down into weeks and topics. It's important to just point this out, that each lecture obviously has its own learning outcomes, and these will frame what you need to know and extract from that module. These will be shown at the beginning of each lecture, but they'll also be placed on Canvas as well if you want to look at them there. Also on the module homepage, we have quite detailed reading lists. So you can find your book chapters that are associated with what you need to know. Also some case studies and journal articles. But also there'll be some additional resources, some videos and links and a variety of things like that. Also, we've got the discussion boards. So please, if you've got a question, it would make sense if you kind of ask it on the discussion board first, because someone else may be able to help you out, but it may also be that a similar student has the same kind of question. So therefore, rather than immediately asking me, if you put it on the Canvas board, then we can address it and it can be made available to all students. And so that will sort of help everybody out. Also, during the lectures, quite often we make use of Poll Everywhere. So if you don't already have the app on the phone, it's kind of good to get it as well, but you can just use it through the web links as well. So this should be sort of your point of call for anything you need to know about the module. So please do make use of the resources that are available on there. And then in terms of reading, I put these books up here. So Kubi Immunology and Janeway Immunology are the sort of core texts that are associated with the immunology side of this course. I have also put on here this male immunology. It was also known as Roy Brostoff and male, so that's kind of like a good introduction into the topic. But really, Kubi and Janeway immunobiology are the core texts, and you can use either one of these. So in terms of the reading list, we'll put the page numbers for each of them. The Kubi immunology is available in a digital copy in the library. The Janeway one is only a few copies of this, actual physical copies. Yeah, and then of course we're monitoring the attendance, so let's do that now. So the pin for the lecture is 1979. That's 1979. Anyone need the pin again? You've all got it? All right, so we'll start now with sort of our introduction into immunology and the immune system. So these are the learning outcomes from today, but also for this week. So you'll be familiar with the common terms that we use in immunology and also the key definitions. You'll be able to understand the defining features of the components of the immune system. So this will include various barriers, such as the physical, chemical, and microbial barriers, but also the innate and the adaptive immune responses, the variety of different cells and mediators that are involved, as well as various lymphatic organs and circulatory systems. You'll be able to understand that the immune system can distinguish between self and non-self. Also that the immune system recognizes what we call antigens on pathogens, and it does this via specific receptors on immune cells, but also to describe the properties that distinguish the innate and adaptive immune response. So first of all, how does the immune system work? And this is where I'd like you to just take a couple of minutes to just do this on poll everywhere. So when you hear someone say the immune system, what do you think of? There's no right or wrong answer here. It can be anything you want. So you can either scan this QR code here or go to poll everywhere and then just put in Wood David as the code. Write as many words and phrases as you want, and then we'll have a look at that in a couple of minutes. All right, so that's actually looking like you guys have got a really good understanding of it. It's obviously great to see things like antibodies, pathogens, vaccines and vaccinations as well, protection, phagocytes. Autoimmune dysfunction, the memory response as well. Well, that's perfect. And some emojis as well. All right, that's brilliant. That's a really good start. So let's switch back to the lecture. So obviously, if we think about the immune system in terms of your life and everyday understanding, COVID-19 obviously really brought sort of immunology and the immune system to the forefront in terms of the public knowledge, because obviously everyone was interested in the immune system, but also vaccination and how quickly we could develop a vaccination. And in some respects, immunology is quite a sort of modern discipline. It arose out of microbiology, but it has been around for quite a long time, and certainly some of its roots date back many, many sort of centuries. And you can find that kind of information on the web and things like that. But obviously, recently, COVID-19 brought our attention to immunology. But if we look back at some of the major advances, we can see that obviously the eradication of smallpox via vaccinations in the 1980s was obviously a major success and was probably one of the key defining moments for immunology. We also know that the immune system is really important because individuals that actually lack an immune response, such as this boy here, who's David Vetter, who was born in the 1970s, he actually lacked an adaptive immune response, and he actually had to live his life in a bubble, in a plastic bubble. Sadly, he died when they tried to correct his immune dysfunction by giving him a stem cell transplant that was actually infected with EBV, and that caused him to get lymphoma. But we know that the immune system is vitally important. Also, there's links between inflammation and depression. So inflammation is actually one of the key features of the immune response. And that's now coming to the forefront as well, this kind of link between depression. And then I've put up here this slide of chickenpox, because obviously you know that as a child or an adolescent you get chickenpox, but then hopefully that's the only time you'll get it. And the reason that you'll only get it once is because you've got an immunological memory. So, that's another key feature of the immune response, is that it will provide you with a sort of memory response and protect you, hopefully, for life against various diseases. It also plays an important role in cancer surveillance. So every day, your immune system will be eliminating transformed cells and preventing them from developing into cancer. So that's a vital function there. But also, it offers the sort of promise that if we can sort of genetically engineer some of our immune cells, we can use this for providing a personalized treatment against cancer for various patients. And then we have this here. So does anyone know what this is? So we've got some syringes that are filled with a rather strange-looking brown liquid. Anyone want to hazard a guess at what that is? Anyone? It's actually poo. So we're thinking here about fecal transplants, and the link here is the microbiome. So obviously, in recent decades, there's a lot of focus has been put on the microbiome. So the microbiome, primarily sort of, it's all throughout your body, but obviously there's a large load of it in the gut. And obviously if you have no immune response, if you have no microbiome, you have no immune response. They're really interlinked, and it plays a key function, and it's really vital for your health. So I just kind of wanted to link that there, because obviously some individuals that have some severe complications, they can actually be rectified by sort of giving them a healthy microbiome via a fecal transplant. So we continue to look at the immune system. We know that it's vital to human health. There's lots of things we don't know. It's a constantly evolving field, and in fact, the immunology that I learned at university was much more narrowly focused than it is now. So as research goes on, it constantly is developing. It's full of interconnecting parts, and I put up here some of the family of cells that we'll be looking at. So we have our lymphocytes, our granulocytes, and of course our dendritic cells, which all play important parts. And these are all moving around the body, so it's kind of interconnecting. It's fascinating as well. It's quite complex, but it really is fascinating, and I hope at the end of this module you kind of get an appreciation and a fascination for it too. Because personally, I think it's one of the most interesting aspects of the life sciences. But then, I might be biased. It also integrates with other parts of biology, so there's probably not a physical or mental illness that doesn't have some sort of aspect of the immune system going on within it. And it's also wide-reaching in terms of scientific research. So I put here a Western blot because, if you're doing a Western blot in the lab, you're using antibodies to detect the protein. So actually, many aspects of the immune system are widely used sort of in the lab. And if you do work in a lab, it's pretty certain you will have come across some of these aspects. Even if you don't study actually immunology, they're kind of spread out through all the fields in terms of scientific research. Now, immunology is challenging, and I'll just put up this statement, which is really quite a provocative statement from a former dean of the Harvard Medical School. And he said, half of what you were taught will be wrong in 10 years' time, and the trouble is that none of your teachers know which half. I don't know that I necessarily agree with that, but it kind of reinforces that fact that research is constantly evolving, and as it does, our knowledge evolves and things change, and depends on certain situations. So it's always kind of worth bearing in mind that you need to keep up to date in terms of the literature and things like that, because nothing is static. And the answer to most immunological questions tend to start with, it depends. And that's because it is quite complex, but it depends sort of on what sort of setting you're looking at, and things can have sort of different outcomes depending on whatever setting. So it can be a challenging subject, but like I said before, it is very fascinating. So how does the immune system work? Well, we know that it protects you from infection. It also protects you from cancer and foreign substances such as pollution. It plays a really important role in healing from damage, as well as repair and growth, but also in pregnancy. And that's something that's really kind of quite important. It also plays a really essential role in differentiating between self and non-self. So when we talk about self, we obviously mean our own cell tissues, and non-self perhaps being a pathogen or something like that. But then that also applies to healthy versus non-healthy. So as we mentioned earlier, the immune system plays a really key role in actually detecting cancer and transformed cells. So that's how that would fit in there. And then the immune system, it is an umbrella term because it has multiple components and systems sort of within it. Now, when it comes to the immune system, it is a little bit of a balancing act. So on one hand, we have the need to kill the pathogen, but we also don't want to harm the host. So in order to kill the pathogen, we need to create a hostile environment for that pathogen. But you can imagine that if we go a little bit too far and we make it too hostile, we'll kind of create damage to the host cell tissues as well. So it's a really fine balance between creating a hostile environment to kill the pathogen, but not actually harming the host. But it doesn't always work. So sometimes the immune system isn't successful in eliminating that pathogen, and we can't actually sort of eliminate that infection, which is obviously problematic. But on the other hand, the immune system can sometimes go too far, and it can harm the host. And an example of this might be autoimmunity, where the immune system is actually targeting our own body cells and causing us issues. So it really is kind of a fine balance. And it's very important to remember that the immune system is constantly changing throughout our lifetimes. So it's dynamic, constantly adapting and changing. As we discussed in the last slide, we know that it's protecting us from harm from the environment, so this includes pathogens and chemicals as well as damage. It's also playing a really key role in recovery from illness and injury. But it will actually vary throughout a lifetime. So when you're born, you're very much reliant on the maternal antibodies that you've got from your mother, and that will sort of see you throughout the first 6 to 9 months of life before your immune system starts to kick in. And then obviously when you're a toddler, you're getting lots of those sort of infections that are really shaping and honing your immune system. And then, as I mentioned before, we have some changes during pregnancy. Obviously, you need to protect the fetus from the mother's immune system. So, there's changes there. And then, as we age, the immune system will steadily decline. The thymus actually shrinks, and the immune system will start to sort of ramp down as we age. So it's not static. It's always changing, and it will change throughout an individual's lifetime. And it's unique. We probably all know that one person who never seems to get a cold or a sickness when everyone else around them is getting one. And again, that's due to sort of the immune system. And we're kind of, if you stick with this and you do immunology in the third year, immunology in health and disease, we'll kind of go into that in a little bit more detail. But the immune system is unique and it differs from person to person. Now, immunology, as I've already said, is quite a complex led network. And this does give some challenges in learning immunology, but also teaching immunology. So it's this kind of complex led network of cells. Now, these are mostly white blood cells, but they're not all necessarily located in the blood, so some will be in the peripheral tissues. And as we discussed earlier, they're quite mobile, they're not fixed in one location. We also have a variety of different molecules that play key roles. So we have antibodies, cytokines, and signaling molecules. We also have involvement of a variety of different organs. So you'll have your bone marrow, where the cells develop. We have spleen and the thymus as well. And then we also have circulatory systems, such as the blood and the lymphatics, throughout which the cells move around our bodies. And then we have a variety of tissues and barriers. So these would be the skin, the gut, the mucosa, and various other associated barriers and tissues. So we look at that in terms of the layers of immunity. So you can imagine here that these represent viruses that want to get into our body. The first barrier that we're going to come across is obviously the physical barriers. So our skin, hair or fingernails, things like that. If the physical barriers are breached, we then have the innate immune system. So the innate immune system is ready to go. It's very quick, but it's non-specific, okay? So this will kind of kick in very quickly. Should that fail, we then have the adaptive immune response. So the adaptive immune response is slower to get going, but it's specific. And it's the adaptive immune response that will give you the memory that we kind of need from being exposed to a variety of pathogens and things like that. So they're the kind of three barriers, and hopefully by the end of it, the adaptive immune response is kind of like the big guns of the immune system. So if a pathogen gets past the physical barriers of the innate immune system, the big guns or the adaptive immunity will hopefully clear it out. So we look at the layers of defense, and so this is kind of looking at the gut mucosa as an example. We have a variety of sort of different layers of protection and a variety of different checkpoints. So one of the first barriers that we might have is our microbiome, so our microbiological barrier. You can imagine that obviously if we have healthy commensal bacteria, that kind of prevents pathogenic bacteria from getting a foothold. We also have physical barriers, so we'll have our epithelial cells with the tight junctions, kind of knitting them together. We also have a layer of mucus that provides protection. And in the gut, you also have actually secretory antibodies as well that play an important function. If these physical barriers are breached, we have chemical barriers. So an example of this will be the complement system that we'll talk about in the next coming weeks. We then have the innate immune cells from the innate immune system. And should these fail, we have the adaptive immune cells. And the actions of these are all kind of coordinated through molecules that we call cytokines, so signaling molecules. So they're kind of talking to each other. They're not operating independently, and that's a key thing to actually keep in your mind. In terms of the barriers, this diagram is just showing you some of the strong barriers. So in blue, it's highlighting hair, skin, epithelium, as well as fingernails. So these are our really kind of strong, robust barriers. And then in red, it's showing you vulnerable barriers. So these will be your mucosa. So, you can see we've got our oral cavities, our lungs, so the respiratory tract, the urogenital tract, as well as the GI tract as well. So these are all somewhat vulnerable and need protection from the immune system. And then this is just showing it in a little bit more detail. So we're looking here at the skin, the gut, the lungs, and then the eye, nose, and mouth. And we can kind of divide it into mechanical, chemical, and microbiological. So if you think about mechanical, for instance, in the skin, the gut, we have this kind of flow of air or fluid which is helping to remove any pathogens. In the lungs, we have cilia, so these are obviously, you know what the cilia are, but they kind of beat away. So we've got the action of mucus and cilia, which is kind of removing and beating away any pathogens that may sort of come in to the lungs. And then we have tears of the eyes that will kind of flush any pathogens away. In terms of chemical barriers, we have fatty acids on the skin. And then in the gut, we have low pH, obviously, so that will destroy most pathogens that kind of enter through the mouth. The lungs, we have surfactant, which helps. And then we also have sort of antimicrobial enzymes in the tears, but also we can find them in the gut as well. And that plays a really important role. And then we also have these antimicrobial peptides, which are found across all of the barriers that help to make kind of a hostile environment for pathogens. And then we have our microbiome. So as we mentioned, that plays a really important role. So these will be our commensal bacteria, our healthy bacteria, our friendly bacteria, if you were, that also plays a really key response. But it's important to remember that obviously the barriers vary with each location, and they're quite distinct. So if we look at the barriers of the skin, you're obviously aware that this is multiple layers thick. So it's providing protection in that way. It's watertight. And then we also have our chemical barriers. So as we mentioned, we have pH, we have enzymes, as well as antimicrobial peptides, all helping to kind of maintain that barrier. If we compare that with the lung mucosa, We can see that this is obviously very thin. It's only one cell thick, so it's obviously quite vulnerable in that respect. But we've got a protecting layer of mucus as well as cilia to sort of sweep away any pathogens that enter there. And then in terms of the gut, this is probably the most complicated of our actual physical barriers because it's constantly changing throughout the entire length of the gut. In the small intestine and the large intestine, the physiology is actually quite distinct and quite different. But you can see here, we've obviously got our microbiome, so our protective microbiome layer, and then we've got a layer of mucus as well, and we have quite specialized cells that are all playing key roles in sort of maintaining that barrier function. So let's have a quick touch on the microbiome. As we've already mentioned, the microbiome is really important. So on the left-hand side of the diagram here, you can see a healthy colon, which is colonized by quite a large number of commensal bacteria, so our friendly bacteria. Now, let's say you have an ear infection and the doctor puts you on antibiotics. That's great. It's going to clear the ear infection, but it's also going to have a rather devastating effect on your gut, as you can imagine. Now, what happens with antibiotics? You can see that the number of commensal, our friendly bacteria, really decreases. This actually sets, a space for potential pathogenic bacteria to come in and get a foothold. And if these are releasing toxins, you can imagine that that's obviously going to have an effect on your quite fragile mucosal layer, which is only one cell thick. And then it starts to get breached. It becomes a little bit leaky, and we kind of get the contents of the gut entering into the bloodstream. And obviously that's a really bad situation to be in. And that can obviously lead to disease. So that's why it's really important not to overuse antibiotics, because you want to maintain your microbiome. And by maintaining your microbiome, that helps you to maintain a healthy physical barrier. Now, when it comes to pathogens, different pathogens really require a different type of immune response. So it's important to think about the immune response, or not just from the body's side or the immune system side, but also you want to think of it from the pathogen side as well, or the pathogen's perspective. Because you can imagine some pathogens are extracellular and some are intracellular, and obviously they require a different type of immune response. So if we have an extracellular pathogen, this can be easily accessible to the soluble parts of the immune system and the soluble molecules, so it can be targeted quite easily. But if we have a pathogen that's hiding out intracellularly, that's kind of not so easily accessible to the immune system. And if we want to destroy it, we actually have to kill that infected cell in order to make, to eliminate it, basically. So you need to think whether we've got an extracellular pathogen or an intracellular pathogen, and then we can tailor the immune response accordingly. So let's have a look now at some of the chemical barriers. Some of these we'll talk about in the upcoming lectures as well. So we have saliva, tears, and the stomach acid, so the low pH that we talked about earlier. And then we also have some sort of immune-specific chemicals as well. So these can be antimicrobial enzymes, antimicrobial peptides, cytokines that we'll talk about in the future, and then acute phase proteins and the acute phase response, which we'll also talk about in the future, and the complement system as well. But we'll just have a look at a couple of these today. So if we have a look at some of the antimicrobial enzymes, a good one of these is lysozyme. And this is actually playing a really important role in breaking down the peptidoglycan of the cell wall. And this works quite efficiently in gram-positive bacteria. So you can see the structure here of gram-positive compared to gram-negative. So the gram-positive have this sort of exposed peptidoglycan layer. And we have lysozyme as this little sort of yellow Pac-Man. And what happens is the lysozyme will come in and it will break down and digest away the peptidoglycan, and that will expose the lipid bilayer and therefore make the pathogen vulnerable. So that plays a key role there, and that's secreted in saliva and tears as well. If we look now at antimicrobial peptides, so these are known as AMPs, and this is kind of an umbrella term for defensins, cathelicidins, and histatins. And these are activated by proteolysis. But they all have their sort of own unique function. So the defensins play a really good role at actually penetrating and breaking into the microbial membranes, and that's what is being shown in this diagram here. So they disrupt the integrity of the microbial membrane, and these can be released by various immune cells, so neutrophils and macrophages, which we will talk about in the upcoming lectures. But you can see here defensin, it kind of forms this pore in the microbial membrane, and obviously if you form a pore, then that has pretty dire consequences for the pathogen. We have cathelicidins, so again these are produced by neutrophils as well as various epithelial cells in response to infection, and they are able to neutralize endotoxin, but they also have antimicrobial toxicity. And then we have the histatins, so these are primarily produced in the mouth. So you may know that if you get a cut or a burn in your mouth, it heals pretty quickly compared to like a cut on the skin. And that's actually down to the role of histatins. So they promote this rapid healing and repair of the oral cavity. But they're also really good at fighting pathogenic fungi. So Candida albicans as an example there. So they're really sort of key players as well in the chemical barrier. And then, as we mentioned, immunity, it can be innate or adaptive, but it can also be humoral or cell-mediated. So humoral basically means secreted or soluble factors. So, you know, we might think here about antibodies because they're secreted and soluble. So antibodies are secreted by B cells or B lymphocytes. And then we have the cell-mediated arm. So cell-mediated is involving your white blood cells. So, for instance, T cells, T lymphocytes. And these will directly eradicate pathogens as well as target perhaps transformed self cells, and they basically play a key role, and that's just shown in the diagram here, that we can have a humoral response as well as a cell-mediated response. So I want to bring you back to the innate and the adaptive immune response. So as you remember, we have our physical barriers. Now physical barriers will actually be sort of, you know, they do come under the innate immune response as well. But if our barriers are breached, then we have our first line defense, which is the innate immune response. Now, if that is breached, we then, as I mentioned earlier, we then have our full-on big guns, so our adaptive immune response, which is, again, delayed, but it is delayed because it is specific. So it takes that time to kind of ramp up and produce a specific immune response. So the immune cells that play a key role in the immune system are our white blood cells, and these are obviously our leukocytes. Now these can be found in the blood, but also the peripheral tissues, as well as various specialized lymphoid organs. So we have our central lymphoid tissues. These will include the bone marrow and the thymus. So for instance, T lymphocytes will actually, they'll originate in the bone marrow, but then they'll move to the thymus where they undergo their development. And the function of these is actually to support the development of the cells that are involved in the immune system. We then have our peripheral lymphoid tissues. So this will be our spleen, adenoids, the appendix, the tonsils, our lymph nodes. And then we also have sort of these less organized lymphoid tissues. So these will be your mucosal-associated lymphoid tissue. And we find these, for instance, in the gut, we have the gut-associated lymphoid tissue. And basically these sort of, you know, they trap antigen and they initiate the immune responses. And hopefully you're familiar with this diagram. So this is just showing you hematopoiesis. So at the top here we have our multipotent hematopoietic stem cell. And this gives rise to all the cellular elements of the blood. So this includes all the cells of the immune system as well. And this multipotent hematopoietic stem cell can be divided into two types of stem cells. We have our common myeloid progenitor and then our common lymphoid progenitor. So the common myeloid progenitor will give rise to erythrocytes, but it will also give rise to cells like mast cells, basophils, neutrophils, eosinophils, and monocytes as well. And then we have the common lymphoid progenitor, which will give rise to our lymphocytes, so our T cells and our B cells, and also natural killer cells. And it's really important to remember that obviously leukocytes is the collective term for white blood cells, but not to confuse it with lymphocytes. Okay? So leukocytes are white blood cells. Lymphocytes are a class of leukocyte. And so these are our lymphocytes here, our T cells and our B cells. It's also quite helpful to look at this hematopoiesis in terms of the innate and the adaptive immune response. So shown in this sort of yellow box here, we have the cells of our adaptive immune response that are coming from the common lymphoid progenitor, and these are our lymphocytes. So essentially, our T cells and our B cells. So it's quite easy to remember in that respect. And then over on this side, we have our innate immune system. So our mast cells, basophils, neutrophils, eosinophils, and monocytes. Now, one other thing to point out is that the adaptive immune cells can actually differentiate. So that's what you can see here. So in terms of the lymphocytes, we have our T cells and our B cells, but B cells can differentiate into plasma cells that secrete antibodies. And then we also have our T cells that can differentiate into a variety of different cells. So we have our T helper cells, or our Th1, Th2, Th17, 9, cytotoxic T cells and Tregs, whereas our innate immune cells don't do that. We have one class and they don't differentiate, okay? So a basophil is a basophil. Now it's important to become familiar with these cells. So here we're looking at a blood smear. And we've got our blood cells, so our erythrocytes, red blood cells here, but we've also got neutrophil, eosinophil, lymphocyte, monocyte, and basophil. So you can see that they have a distinct appearance that obviously can help to identify them under the microscope. And they're also present in our blood in different proportions. So you can see that a neutrophil will account between 40 to 70%, whereas a lymphocyte, may account between 20 and 50%, and some of the others are quite small in proportion. And this is quite useful, being able to identify them on a blood smear under the microscope, because you can then determine whether an individual's immune system is within the normal range, whether that individual is healthy, or perhaps whether that individual is suffering from an infection, by the different proportions of the cells that are available or can be seen in a blood smear. So why does the immune system have so many different components? Well, this kind of leads on to what we were talking about earlier, that the immune response is really kind of tailored to the type of organism that is involved, and that may depend sort of on the location of that pathogen as well. So obviously a pathogen is a collective term for any microorganism that causes a disease when it infects the host. So we have helminth parasites, so these will be your worms. We might have protozoa, fungi, extracellular bacteria or intracellular bacteria, and viruses. So you can imagine that the immune response that targets an extracellular bacteria is going to require some different functions to one that, if you have an intracellular pathogen or anything like that. So the immune response is tailored depending on the pathogen that you're infected with. If we look at inflammation, so inflammation is something that we'll cover in the next few lectures. But inflammation is essentially a hallmark of the immune system at work. So you can see here we get a small prick on the finger. This introduces some bacteria. Once we get bacteria, this will activate our resident effector immune cells. These will release cytokines, so signaling molecules that will help to recruit additional immune cells. We get this vasodilation, we get permeability, and we kind of get this inflammation and swelling. And what we end up with is all the hallmarks of inflammation. So we get redness, heat, swelling, and pain, and also loss of function. So the immune system, basically inflammation is your immune system at work. And we will discuss that in the next few lectures as well. So let's have a look sort of in terms of the innate and the adaptive immune response. These are the cells from our adaptive immune response. We've got our lymphocytes, so we've got our T lymphocytes, or our T cells, B cells, and then we have the plasma cells, which have differentiated from our B cells. Now, we've already talked that the innate immune response comes first, but we have to really stress the fact that the innate and the adaptive immune response, they're not separate, they're interlinked, and they do communicate and talk to each other, okay? And essentially, the really powerful and specific adaptive immune response will only be switched on via the innate immune response. And there's some cells that play a really key role in this, and one of them is the dendritic cell. So the dendritic cell is this guy here, and here's a professional antigen-presenting cell. And we can also look at that on this diagram, which kind of nicely summarizes it. So we've got innate immune cells here, and then our adaptive immune cells on the right-hand side, and we've got the dendritic cell in the middle. So some of the innate immune cells will actually act as antigen-presenting cells, and they will sort of basically communicate with the adaptive immune response. And these antigen-presenting cells really do play a really crucial sort of link between the innate and the adaptive immune response. So here we have the dendritic cell eating a bit of a pathogen, and then we have him presenting it to the T cell over here. So it's really important to remember that the innate immune response kind of directs the adaptive immune response. And this diagram is also quite useful in showing that. So here we're looking at an immune response over time. So we've got the initial infection over here on the left, and hopefully what you can see is we've got the innate immune response in blue, and this will ramp up pretty quickly after infection. And this will hopefully deal with it. But if the innate immune system doesn't actually deal with the infection, then we need to bring in the adaptive immune response. And so you see the adaptive immune response is slower, it takes more time, but it is specific. So whilst the innate immune response is immediate and non-specific, the adaptive immune response is specific and will give us that memory function that we so need, really, in order to provide us with that memory function. And then obviously once the infection is cleared, the adaptive immune response ramps back down and we go to our steady state. And so this just summarizes what we've been going over today, that we have these kind of different layers of defense that will help to protect us against the pathogens. We have our anatomical barriers, so these can be our physical barriers, such as the skin, the mucosa, also our microbiome, as well as some chemical barriers. We then have our immediate innate immune system, which is pretty much, it's ready for action at all times. So these will be our chemical barriers, such as the complement system or the antimicrobial peptides or enzymes. If these fail, we have the induced innate immunity, which is mobilized upon the detection of the pathogen. And then we have our adaptive immune response, which is slow, but it will give us a specific and a memory function as well. So in terms of reading, I put up some chapters from Kubi Immunology and Janeway Immunology. You can pick whichever book you choose. They're both really good. And then there's some additional reading down here. But otherwise, I will see you guys on Thursday.